Hello everyone, this is Rahul Bhatia from team Decipher IAS, and today we are going to start a new lecture in Science and Technology series, and the topic of the lecture is Neurons Optogenetics Clarity. Neurons, as we know, these are the cells present in our brain, and optogenetic and clarity are the techniques to study these neurons and how they are arranged in the brain and how do they work. So today we are going to deal with that. So this lecture is important for prelims as well as mains because we get to know something about neurons, which is important for prelims, and we get to know about the techniques which are important for main science and technology portion. So moving forward, so why I have chosen this topic is because recently Professor Carl Desroth, he is a professor in Stanford University. He is also a doctor and also a PhD. So he is doctor, MBBS doctor also and PhD doctor also. So he is a double doctor. So he has just received Life Sciences Breakthrough Prize for 2016. This prize was instituted by Silicon Valley entrepreneurs, that is the big IT firms have instituted this prize to honor the people who devote their lifetime in research in life sciences, in fundamental physics and mathematics. So this becomes your pre-question. Okay, so you have to remember that. So this is the quote by this professor who has received the award. What he says: the suffering of mentally ill and the mysteries of the brain are so deep that to make progress we have to take big risks and blind leaps. This means that it has been said that the brain is the most complex thing in the universe that we are trying to understand. So he is saying that we have to take big risks, means we might have to experiment on humans too, and blind leaps means we have to try different methods, and out of hundreds of methods one can be of some possible use in future. Okay, so that we have to remember. So he is the scientist who has developed these techniques of optogenetics and clarity. So moving forward, what are these techniques? Now I am going to explain. But before that, we are going to deal with neuron. So neuron, everybody knows. This is the structure of neuron, and this is the basic cell which is present in our brain, as well as our spinal cord and peripheral nervous system. So starting here, these are the dendrites. You see like the branches of tree. These are the dendrites. What are their functions? These neurons will be connected with some other neuron, because these neurons are connected with each other to form a circuitry in the brain. Okay, these form the circuits. These are like wiring that we have in our homes. One wire is connected with the other through different circuits. Okay, so this is the part of circuitry. Here this neurons will be connected to the one neuron, and here also it will connected to next neuron. Okay, so what is the function of neuron? The neuron's main function is the transmission of signals. So how the transmission happens? It happens with the help of chemical as well as electrical signals. Chemical is mainly by neurotransmitter, and electrical is by moving in and out of ions. Sodium, potassium ions are there, that I will tell just now. Wait, sodium, potassium ions are there, and which go inside the cell and outside the cell. That leads to change in the electric potential of the membrane, and hence the transmission of the signal. Here this is the cell body or soma, and inside the soma, this is the cell body, and inside the soma this is the nucleus. So inside the soma we have nucleus, then we have axon. This axon is the wire where the transmission of signal happens like this, and it is transmitted to the next neuron. So here axon is covered by this thick sheath. This is myelin sheath. This myelin sheath is made up of lipids. This is just to avoid the interconnection between different neurons, because this is insulating. This acts as an insulator, thereby preventing the contact between neurons, and mixing up of signals is prevented. Mixing up is prevented. So we prevent the mixing of signals by that. And then after the signals move here, it moves to next neuron, and thus conduction of the nerve impulse happens. So moving forward, neurons signal to other cells to fibers called axons. This you see is the axon fiber. Complete fiber is the axon fiber. I already told about neurotransmitters, and these places, this is the place is called synapse. The chemical is moved out through the budding channels, and then it is transmitted to the next neuron, which gets excited, and thus transmissions happens. Now moving forward, neurons are fed by glial cells which are present nearby it. Okay, you have to remember, glia is from glue. So this is the circuitry I was talking about. Here you see all the neurons, complex web is there. So this web is not getting deciphered by scientists. So this scientist created new techniques to study this web. As this web is the, if we understand this web, we can understand the brain, and we can understand the diseases. You know, brain is the most complex thing. It is the wiring between 80 billion neurons. 80 billion. In such a small volume, we have 80 billion neurons interconnected with each other. So imagine it is how difficult to study, and that too because we have lipids. So lipids scatter the photons. They scatter the photons, and hence we are not able to see the inner circuitry of the brain. So moving forward, so because of that, they developed the technique. First one is optogenetics, we are going to discuss. Here they illuminate the brain. Illuminating the brain means they throw the laser light on it, and hence they can modify the electrical activity. How it is done, we will be just explaining, and this is the same thing. This is the explanation, short explanation, which I will be dealing in detail in the next slide. So here what we take is, we take a protein which can be excited by laser, and we take it from alga, and this protein is an ion channel that opens in response to blue light. So blue light excites the ion channel. If ion channel opens, ions move inside, the electric potential changes, and thus we get the transmission of signal, which is also responsible for our behavior, response, etc. Okay, so this, we take this gene, we take the help of gene therapy, whereby we cut this gene and we insert it in neurons, and this expressed on the surface of cell membrane of neuron. And whenever the blue light is put on this leads to excitation. Excitation leads to opening of ion channels, and thus firing of neurons. Firing means opening of channels leading to passing of electrical impulse. Electrical impulse is nothing, just a signal. Electrical impulse. So we can know which neurons are responsible for particular type of response. Okay, response. Now 6 steps are: we make a genetic construct, then we insert into virus. Virus acts as a carrier like a vehicle which transports us from one place to another. It acts as a carrier. Then if virus affects the neurons, this gene gets inserted in the DNA of neuron, and then is expressed as protein. Okay, then we throw a light on it through fiber optic cable plus electrode. Micro electrodes are attached to the brain. Suppose we take a mouse as a sample. I will show you just recently in the next slide. So laser light opens ion channels. We record the behavior. See, this is the genetic construct. We inject in the virus. Virus affects the mice. This is the injecting of virus. Virus affects the mice. Then we have optoelectrode. This is optoelectrode, in which we have fiber optic cable plus electrode. Then we put the laser light here, and then we can check the response of the mice. This is the opsin channel. Opsin is the protein which we took from alga, which is now being expressed. Okay. So moving forward, this is the apparatus. Here we put the laser. This is the mice. This is the light we are putting in the head of mice. So this light leads to change in the response of the mice as it is living. So there is change in the response. We can see it in the monitor with the help of software. So see, this is the excitation of neuron. You are seeing the electric light that is coming out. This is just to show. So now moving forward to the next technique, clarity. Clarity means transparent brain. Means if we can see the inner circuitry. Before this technique was developed, we were not able to see the brain's inner circuitry due to presence of lipids. We have to devise a method by which we can maintain the tissue structure of brain, but we dissolve the lipids. So we have found out this technique of clarity for doing that. So this makes brain transparent by building a hydrogel. We use a hydrogel which removes lipids and remove lipids with the help of detergent, which makes the brain opaque. And now we come to it. We take a hydrogel. Hydrogel forms a mesh. You are seeing a mesh. Hydrogel forms a mesh. You can see the inner cell organelles. These are cell organelles which will dissolve if we dissolve the lipids without creating a mesh. But we put this solution, which is solution of formaldehyde and hydrogel monomer. This leads to formation of mesh binds the tissue. See, hydrogel tissue hybridization. This binds the tissue so that the organelles are at the same place where they were when the lipids were there. But lipids get dissolved. With the help of SDS detergent. SDS detergent dissolves lipids in vesicles and which is washed out. So brain becomes transparent. Here you see. This brain becomes transparent because lipids have washed out. Here you are seeing these vesicles. These are lipid vesicles that I was just telling about. So before, what we see? Before, this was the look of brain. Now see, very blurred you are seeing. This is called transparent brain. So moving forward, see, this is with the help of fluorophores. These are the neurons. Different neurons are shown with the help of fluorophores. These neurons are responsible for different brain activity. So for vision, for hearing, for touch response. Now moving forward. So this is just a simple differentiation that I have just told you. This is kind of a summary. Moving forward. So what are the applications of this technique? Optogenetics and clarity have similar applications. Optogenetics we use laser, so it helps in discovering about how neural circuits control the behavior. Suppose we excite a particular area of brain, how our response is. Then we do the second area, how our response is. So this helps in telling about the response. Other one is health and neurological and psychiatric diseases. I have already told you this is the application. So gene therapy. This gene therapy this is a future vision which we can use to correct the brain diseases or disorders that we have. We can also use it in recovery of vision as seen in mice models. We can also use in finding the cause of Parkinson disease, and also correcting it. Now clarity is the most, because brain is transparent, so we can see the circuit. The other is, it has been found that autism is a disease in children and also in adults. Here we have a loss of memory, so we see the ladder like connections in this, ladder like connection between neurons. Ladder like connections is there between neurons, which is not found in normal brain. So other one is, we can also find the cure of Alzheimer's disease and multiple sclerosis. Alzheimer's disease is person forgets everything. First of all, it starts with short-term memory loss and then hallucinations, and he is in some other world. Okay, so multiple sclerosis is due to dissolution of myelin sheath. Okay, so short circuit happens in the brain, like we have it short circuit in our houses. Short circuit happens, so you become kind of mad, though I should not use that, but you become mentally disabled. So thank you for watching this lecture. I hope you would have liked this lecture. Please like, comment and subscribe to www.decipherias.com. Please like our Facebook page and do comment below in the comment section. Your comments are very important to motivate us to do hard work for you. Thank you.